Good afternoon, everyone, and welcome to the Office of the Chief Scientist Science Seminar Series. For those of you who don't know me, my name is Vanessa Swarbrick, and I'm a Senior Science Advisor with the OCS, and I'll be your host for this year's fall seminars. In the spirit of reconciliation, I want to respectfully acknowledge that our OCS offices are located on the traditional lands and gathering place for Treaties 6 and 7, as well as Region 3 and 4 of the Métis Nation of Alberta. These lands are the homes to diverse Indigenous people, including Cree, Blackfoot, Nakota Sioux, Iroquois, Dene, Ojibwe, Saulteaux, Anishinaabe, Métis, and Inuit peoples, and many others whose histories, languages, and cultures continue to influence our vibrant communities today. Wherever you find yourself today, I encourage you to recognize and acknowledge the relationships that First Nations, Inuit, and Métis people have with the land upon which we all live, work, and learn. In keeping with the tradition of gathering, these science seminars provide us an opportunity to get together, to exchange knowledge on interesting science happening across Alberta in an open learning and sharing environment. The hope is that through our dialogue today, that we are all able to improve our awareness and understanding and to put this knowledge into practice to support evidence-informed decision-making wherever that's occurring. So with that, I'd like to welcome Dr. Steve Wiseman. Steve is an associate professor and Canada Research Chair in the Department of Biological Sciences at the University of Lethbridge. He grew up in Newfoundland, completed a BSc at Mount Allison University, his master's and PhDs at the University of Waterloo, and a postdoctoral fellowship at the University of Saskatchewan. Steve's an aquatic ecotoxicologist. His research aims to elucidate the mechanisms by which environmental chemicals cause adverse effects on aquatic organisms, especially the reproductive performance of fish. His research explores the effects of a variety of different chemicals, including PAHs, plastics and associated chemicals, flame retardants, and 6-PPD-quinone, which is what you're going to hear about today. And so with that, I'm going to pass the microphone over to Steve.
Well, first, thanks for having me here and allowing me to talk. And thank you for everyone for showing up to listen to me. I recognize a few names from the list, and I think some of you have probably heard this talk already. I know Andreas Lewick is on, has heard it many times. So Andreas, feel free to do something else while you pretend to be listening to me. So I'm going to start out, I'm going to show a little video here. So this is a male coho salmon, and this is in the Pacific Northwest. And it is suffering from something called urban runoff mortality syndrome. You can see him, he's sort of gasping for air at the water surface there. So I'm going to talk a little bit about this chemical 6-PPD-quinone, and I'll relate that to urban runoff mortality syndrome. And this is a little bit of work we started doing with support from the Office of the Chief Scientist. But before I get into that really, it's a small amount of data we've collected in Alberta. I want to set the scene a little bit for everyone. So what I showed you was urban runoff mortality syndrome. That was a coho salmon that was dying in a small creek in the Pacific Northwest in the area of Puget Sound. And if you from the video, you can see that fish displaying, mouth gaping, looking sort of loss of equilibrium. I was disoriented in the water column. And this has been something that's been observed for quite some time in the Pacific Northwest. Lots of papers in the literature on urban runoff mortality syndrome, looking at species that might be suffering from this particular occurrence. And a lot of effort went into trying to figure out what was causing it. And it took quite a bit of time. And, over time, they looked at the usual suspects that we look at in toxicology. They looked at things like heavy metals. They looked at things like pesticides. And they could really never figure out what was happening. But through some modeling, what it turned out was that every time you got a rain event, you would see this. And it always seemed to occur in areas near roadways. And there always seemed to be tire wear particles in the, entering the water through this urban runoff that you get after a rain event. And then a couple of years ago, in a study that was published in the journal Science, and for anyone that does ecotoxicology, we do not get to publish in the journal Science very often. When I was a postdoc, we submitted a paper to Science and we got rejected, I think, within 5 hours, probably a record. So this paper got published in Science, which obviously it's a big deal. And in it, they identified the chemical that caused this urban runoff mortality syndrome. This is a chemical that was causing the death of coho salmon in these small creeks following rain events. And the chemical is something called 6-PPD-quinone. And many of you have probably heard of this chemical. It's got a lot of attention in the media in the last year and a half since this paper came out. I won't walk through exactly how or all the steps and how they identified it, but essentially it started off with rubber tire chemical because they had some evidence that whenever you had this urban runoff, you also had high concentrations of rubber tire particle. And by extracting all the chemicals out of rubber tire and going through a series of steps where they fractionate it into fractions with different properties and toxicity testing, they identified this particular chemical as the cause of urban runoff mortality syndrome, so 6-PPD-quinone. Now, 6-PPD-quinone is not actually added to tires. It's a metabolite of a chemical that is added called 6-PPD. And 6-PPD is an antioxidant. And there's many antioxidants that are added into tires. And what they do is 6-PPD, the parent compound, and other compounds, they diffuse to the rubber tire surface as you get wear and tear of your tire, and they react with ozone. So they scavenge ozone, and that helps to protect the tire from accelerated breakdown. When it reacts with ozone, that's when you get the formation of 6-PPD-quinone. So you can see the two chemicals are shown here. They're very similar. After the reaction with ozone, you get some oxygen groups added onto 6-PPD to give you 6-PPD-quinone. I'm sure many of you have seen tires before that have a little bit of red on them. That's actually 6-PPD-quinone you're seeing there. If we collect the bile out of some fish species, we can actually see that it's quite red because they're accumulating 6-PPD-quinone in our exposures. So 6-PPD is intentionally added to tires. It has a benefit. It helps protect it from, or it helps prevent, accelerated breakdown of the tire, which is obviously, what you want when you're driving your vehicle. So this paper came out identifying 6-PPD-quinone as the cause of urban runoff mortality syndrome. And at that time, or shortly after, Department of Fisheries and Oceans put out a call for proposals to look at toxicity of 6-PPD-quinone. So along with my colleagues at the University of Saskatchewan, in particular Marcus Brinkman, who is part of the work I'll show you a little later, we got funded to look at 6-PPD-quinone and its effects on species of fish you find in Canada, and with a focus on things that we find in Western Canada, particularly some salmonids that we find here in Alberta. So when we got funded, before we had the agreement in place, we sort of opportunistically did some work with some fish that we had available. So the University of Saskatchewan, they had rainbow trout, Arctic char, and white sturgeon in their facility. And I was able to go to Allison Creek out near Blairmore here in southern Alberta and get brook trout. So we brought these species back to the lab, acclimated them for a while, and then we initiated some exposures. So my students and technicians started this exposure one morning while I was taking my kid to school. And when I got into my office, I got a text message from one of the technicians downstairs in our aquatic facility. He's like, "You got to come down. We just need to see you for a second." And I thought maybe something was going wrong. And when I walked in our aquatic facility, there were brook trout floating at the surface of the water that were dead. And they had not had this exposure going very long, maybe an hour or 2 hours. So immediately we knew brook trout were quite sensitive to 6-PPD-quinone. And this was only the second species identified as being, very sensitive. So I called Marcus and colleagues at the University of Saskatchewan, and I know they had started their rainbow trout work. And I said, our brook trout are already dying. Well, at this point, none of their rainbow trout were, but eventually they started to die. And so what we had is brook trout are very sensitive So you can see here on the right, I have written a concentration of 0.59 micrograms per liter. That's the concentration that kills about 50% of brook trout. Almost all our brook trout die within 24 hours. Rainbow trout, and this is subadults we did this with, it's about 4 times higher, 3 times higher, almost 2 micrograms per liter that kills 50% of the animals in an exposure. And, you know, depending on the concentration, they can live for up to about 60 hours. But both species are sensitive. So since that work, and that was probably I think that was the second paper ever showing a sensitive species of fish to 6-PPD-quinone. Since that, there's been, you know, a mad rush to identify species of fish that are sensitive and those that are not. And this is not a complete list on this slide, but there's some interesting things that show up. If you look at what's sensitive, there's the brook trout and the rainbow trout from the work that we've done, the coho salmon in the original studies, white spotted char. There's a publication on that, and then colleagues at the University of Saskatchewan are working on lake trout, and they're quite sensitive as well. But then you look at some of the other species that are tolerant to 6-PPD-quinone, and that's not a complete list, but what I'm showing are most of the salmonids. And some of this is not published work yet, but things like the Chinook salmon are tolerant. Atlantic salmon, for the most part, are tolerant, although there seems to be a caveat here where there might be some sensitivity at specific life stages, and that's some work that colleagues in New Brunswick at Huntsman Marine Science are doing now, but they haven't published that. West Slope cutthroat trout, thankfully, tolerant. Brown trout are tolerant. Bull trout, some work that we did with DFO in Winnipeg last summer, thanks to Andreas for putting me in contact with a DFO team there that had bull trout. They seem to be tolerant. The really interesting one to me is Arctic char. Arctic char, if you know the species name, it's Salvelinus alpinus. Well, they're tolerant. Brook trout are also a Salvelinus, but they're the second or third most sensitive species we have. So it's very interesting. Species that are closely related phylogenetically, you know, don't have the same pattern of sensitivity or tolerance to this chemical, and that's fascinating. Is something that we're looking into now actively. So this is that's a rainbow trout up there that's dying when exposed to 2 micrograms per liter of 6-PPD-quinone. And so one of the questions we were working on, and it's not work that we're doing as part of this funding we have from the chief scientist, but some things we're doing as part of our DFO funding, is asking why are fish susceptible and why are there species differences? As to why they're susceptible, it seems to be, and this is probably not a surprise to anyone when you look at particularly that first video I showed, they seem to disrupt respiration at the gill. So the gill seems to be the target of this chemical. It uncouples mitochondrial respiration, so they're left gasping for air. They essentially can't breathe. And then we see massive species differences. So we have this effect in some species, but not others. And it seems to be related to the ability of a given species to metabolize 6-PPD-quinones. This is a paper we have right now in preprint. It's been reviewed by the Journal of Environmental Science and Technology, and we're just getting ready to submit the revised version. But in that, we show that species differ in their ability to metabolize 6-PPD-quinone, and it seems to correlate with their sensitivity. So those that are tolerant can metabolize it rapidly. Those that are not tolerant cannot metabolize it. And we're starting to identify some of the metabolites and working towards a better understanding of why certain fish species die and why others don't. So this brings me to Alberta. So as we were doing this work, and we saw these results with brook trout, I think I sent a text message to Andreas and said, "You're not going to believe what's happening. Like these brook trout that were exposed to 6-PPD-quinone are dying in a matter of hours." And so Andreas was really excited, I guess, scientifically, probably not excited for the fish, but excited about this. And he was kind enough to put me in contact with the chief scientist's office. So talked to Andy Paul, talked to Jonathan, and I think the chemical was already on their radar. And so, you know, they were kind enough to fund us to start looking at 6-PPD-quinone in Alberta. Up until now, there had been no measurements of this chemical in Alberta. And with the evidence that, it can be quite potent for certain species of salmonids, and knowing how important some species of salmonids are in Alberta, both, recreationally, economically, and culturally as well. We wanted to start looking into concentrations. So we picked, we started to collaborate with EPCOR in Edmonton to do some sampling for us, the city of Calgary, and didn't have that connection here in Lethbridge, but I had a technician, Emily Mertens, in my lab, and she did the sampling here in Lethbridge. And what we did was we sampled some selected stormwater sites in each city. In Calgary and in Edmonton, they have auto samplers. So those are triggered after a rainfall event, and they capture stormwater entering into the river. In Lethbridge, Emily bought a raincoat and some rain boots, and she went out with some buckets and collected water, a little less sophisticated than in Edmonton and Calgary. So there are massive differences in the sampling protocol. But we would tend to go out within a couple hours after a rainfall event and try and sample. So this is not an apples to apples comparison across each of these centers. That's important when interpreting some of the results. And I'll say some of our interpretation is still ongoing. So some of you will be familiar with the sites that I've listed here. For example, in Edmonton, 30th Ave, Cannondale, in Calgary, Anderson, McLeod, Valleyfield. Some of you may be familiar with the areas down here in Lethbridge that we're sampling at. I am not that familiar with the areas, particularly in Edmonton. So we'll just go through a little bit of what we did. We went out and we collect stormwater. Again, in Edmonton and Calgary, it's auto samplers. Edmonton is more of a composite sample over a longer period of time after a rainfall event. Calgary is shortly after rainfall events, and Lethbridge, they're grab samples, usually within 8 hours, actually always within 8 hours, but usually within about 3 to 4 hours of the start of rainfall. We collect those samples, we freeze them, we ship them to University of Saskatchewan, where Marcus Brinkman has an analytical method using a high resolution mass spec method to look for concentration of 6-PPD-quinone in these stormwater samples that we were collecting. And we did these samples over the period of a few months, and the goal was to always go out and collect them after rainfall events. And we have a couple samples during dry periods. Now, there's differences in rainfall events in Edmonton, Calgary, and Lethbridge in the frequency, in the intensity. So again, it's not an apples to apples comparison here. Here in Lethbridge, we go extremely long periods of time without rainfall. And there's differences across our cities. So I'll just walk through briefly. Again, a lot of this is still ongoing. We have data from EPCOR in the city of Calgary on some of the land use in the areas where we've collected samples. We're still inputting that into some of our analysis. The other thing for these graphs I'm going to show you, the scale on the y-axis is not the same on all of them, so just be aware of that. So here's the data for the city of Edmonton. These are samples from May, June, July, August, September, and October across each of our sites. And what you can see, first of all, is that we're detecting 6-PPD-quinone. And that's to be expected. There's roads, there's water, and there's cars with tires. And there's rainfall. So you're going to get 6-PPD-quinone. It's used in tires. Tires have wear and tear. So this is not unexpected. For the most part, the concentrations are quite low and pretty consistent across months at the different sites, but there's a few times there you can see 30th Avenue in September, we get a nice little spike up. At Growit, I think is how you pronounce it. In June, we get a spike in concentration. So we're seeing 6-PPD-quinone, and there's a little bit of variation from site to site and then from month to month. And again, this is all dependent upon the intensity of the rainfalls. And that's part of our analysis, is looking at the rainfall data for these different sampling times. City of Calgary, again, we're detecting it. A little different than Edmonton. You can see here for Valleyfield, we don't have a lot of samples for Valleyfield. We only really had the one sample we were detecting 6-PPD-quinone. Anderson-McLeod seems to be our area where we're getting higher concentrations relative to Britannia, Valleyfield, and Montgomery. But again, we have cars, we have roads, we have rainfall, we have stormwater, and when we're seeing 6-ppd-quinone. Come to Lethbridge, exact same thing. At each of our sites, we're seeing 6-ppd-quinone. We see sometimes, following certain rainfall events, and for example, this rainfall event at Bridge, where you see the higher concentration. That was a pretty intense rainfall, and we got out there a couple hours after. But we're detecting the chemical, and we see variation from site to site and month to month. I like this picture. Some of you may have seen it on my title slide, but this is a picture from Lethbridge here. We're at a stormwater outfall just below the bridge that crosses over the Old Man River. And you can see here a rubber tire actually in the storm wall or in the stormwater outfall. So I thought that was a great picture that sort of captures what we're doing in this project. And I've shared that with a reporter from the CBC who's doing a documentary on 6-ppd-quinone and some of the work that we've been doing in other groups across Canada. So 6-ppd-quinone is present in stormwater in each of our cities, Calgary, Edmonton, and Lethbridge. The highest concentration we saw was in Calgary, up to about 1.6 micrograms per liter. And then in Edmonton and Lethbridge, it's about 0.94, 0.86. Now, again, the intensity of the rainfall events is not exactly the same across all these sites. The method of sampling isn't the same across all these sites, so it's not like Calgary is worse than Edmonton or Lethbridge or anything. It's just that sample that we happened to capture at that time. That's the concentration we had there. But there's a lot more work that needs to be done to really refine this picture of 6-ppd-quinone entering our urban waterways in each of these three cities. But this data clearly shows that it's in our stormwater entering our rivers. So how does this data we have compare to other regions? So this is not all of the data that's out there, but it's some. So in the US West Coast, the areas where they've been seeing this urban runoff mortality syndrome with coho salmon, in stormwater, they see concentrations of about 0.8 to 2.5 micrograms per liter. In Toronto, it's about 0.3 to 2.3 micrograms per liter. In Saskatoon, a study that was done by Marcus and others there, concentrations of about 0.1 to 1.4 micrograms per liter. There's not much data on receiving waters other than in the US West Coast, where you get concentrations up to 3.2 micrograms per liter. Now, just to go back, if you look at the concentrations we're seeing things up to about 1.6 in stormwater in Calgary and around 1 microgram per liter in Lethbridge and Edmonton. So we're in that range of what other studies are seeing in the US West Coast, Saskatoon, and in Toronto. So that's probably to be expected again, cars, roads, rain. So the question then is, okay, we have 6-ppd-quinone in our stormwater. It's entering into our rivers. Is there a threat to resident fish? And that's a big question, and it's going to take a lot of time to go through that. But what I've done is I'm re-showing all the graphs from Edmonton, Calgary, and Lethbridge here, and I've plotted on them the LC50 values. So those are the concentrations that cause 50% mortality in exposure studies. So I've shown here the rainbow trout 96-hour LC50. So that's the study we initially did at the University of Saskatchewan with sub-adult rainbow trout. The brook trout value of 0.6 micrograms per liter. And then another value here for rainbow trout. This is something we haven't published yet, but we're in the process of writing up. This is the rainbow trout larval chronic LC50. So I'll explain what that is. In this study, we took rainbow trout eggs, eyed eggs, and once they hatched, we put them into different concentrations of 6-ppd-quinone, and we grew them out for about 3 to 4 weeks post-hatch. And you can see here the LC50 is about half of what it is in adults. So the earlier life stages are more sensitive than the adults. And that's pretty common for most environmental contaminants, and it holds true for 6-ppd-quinone. And then on the bottom, I have coho salmon, the LC50, and coho salmon being our most sensitive species identified to date. So what you can see is that a lot of our concentrations in stormwater, that's important, this is in stormwater, are around, and some are in excess of that LC50 for coho salmon. And some of our samples have concentrations that are approaching the LC50 for larval rainbow trout and subadult brook trout. So that's in Edmonton. Same thing in Calgary. A lot of the concentrations in stormwater are in excess of the LC50 for coho salmon and approaching or exceeding the LC50s for early life rainbow trout and then subadult brook trout. And in some cases, we have a few that are up there with the LC50 for rainbow trout. Same thing in Calgary or in Lethbridge, concentrations that are near that LC50 for coho, and then some samples that we've collected have concentrations that are near that LC50 for early life rainbow trout or subadult brook trout. So in all three cities, we have concentrations of 6-PPD-quinone in stormwater. So this is not in the river, this is in the stormwater. But we have concentrations that are at, approaching, or slightly in excess of the LC50s for brook trout, subadult rainbow trout, and early life stage rainbow trout. So our concentrations, we would expect that if we were to take that stormwater, that end of pipe stormwater, we'll say, and bring it back to the lab, we might be able to induce lethality in brook trout and early life stage rainbow trout. That's the sort of concentrations we're dealing with in our three cities for these particular samples that we collected. So the question then is, is there a threat to these species, if they were exposed in our rivers? So rainbow trout, for example, in the Bow River in Calgary. So the concentrations in stormwater are high enough to be of concern. However, these are pretty big, waterways, pretty big rivers. So we have dilution. And the first thing you're taught when you take a toxicology course is the solution to pollution is dilution. So we have massive dilution of stormwater in these systems. And I know we have a lot of stormwater entering into, for example, the Bow River or the North Saskatchewan River or the Old Man River here, but we do have significant dilution. So, I've not heard of any reports of acute lethality in these rivers of say, rainbow trout in the Bow River in Calgary. And it's probably because dilution is preventing any acute lethality in the main branch of the river. Are there sublethal effects of low concentration exposures? Not sure. Stormwater inputs are pulse events. So, you get a rainfall, you get stormwater going in for, hours, maybe a couple days, depending upon the rain event. But 6-PPD-quinone tends to flush out quite quickly. So you get these pulse events. Do these pulse exposures, if fish are exposed, have an effect on them? There's some work being done on that, again, at Huntsman Marine Institute in New Brunswick. So we don't know if that's happening. What about small creeks? So on the main branch of the river, we've got a lot of dilution. What about small creeks? Do they receive more stormwater input so that you get higher amounts of chemical in those? I don't know the answer to that, but that would be an area I think worthy of investigation. In the West Coast, when they were seeing these urban runoff mortality syndromes, these are very small creeks relative to the Bow and the Old Man and the North Saskatchewan. So there wasn't nearly the dilution that we're seeing here in Alberta. And in some cases, you're probably getting 50% or higher of stormwater input into these creeks. But what about chronic exposure scenarios? I don't know if everybody can read it, but this table that I took from that paper that was published in Science, this is estimated global annual environmental releases of 6-PPD-quinone through tire wear particle leaching. And you can see, depending upon the amount of 6-PPD-quinone that's formed from the 6-PPD that's in a tire, you can get scenarios where you have 19 million kilograms of 6-PPD-quinone up to almost 95 million kilograms of 6-PPD-quinone per year being released into the environment. And some of that makes it into water. some through via stormwater or direct roadway runoff. 6PPD-quinone is actually showing up in dust samples, so it's not just a water issue. There's a human health issue as well. We're probably inhaling this substance. But tire wear particles are extremely abundant in the environment. There's a paper that came out recently, and I wish I could remember the numbers from it, but of these microplastic and these microparticles that we're finding in the environment, there's estimates that over 50% of it probably comes from tire wear particles in some areas. And those tire wear particles all have 6-PPD and, of course, 6-PPD-quinone in them. So exposure is definitely real in the environment. In an Alberta context, is there sufficient exposure in our waterways to kill some species of fish that might be sensitive? Probably not because of dilution in our larger rivers, but I think we have some more work to do to look at sublethal effects and potential chronic exposure scenarios, particularly chronic exposures that can result from inputs of tire wear particles and those tire wear particles settling out into rivers and potentially creating these long-term exposure scenarios. But a lot of work to do on that end. But I guess, in short, there's 6-PPD-quinone in our stormwater that enters our rivers. That's not surprising. We expected to find that. The concentrations are on par with what you see in other regions of North America and also globally as well. Some studies out of Australia, Japan, and Germany. So we have it. It's there at concentrations we would expect. Probably not causing any acute lethality in species like rainbow trout, but I think there's some more work to do to look at what the exposures are in our river systems and any potential effects that might be caused by long-term exposures to sublethal concentrations of this chemical. And with that, just thank Alberta. I guess it's not Environment and Parks anymore. I should change that. Thanks to the Office of the Chief Scientist. Thanks to Andreas for really getting us in contact with the Office of the Chief Scientist, our partners at EPCOR in the city of Calgary, and then colleagues at the University of Saskatchewan. And my email address is down there, and I'm always happy to receive emails from anyone with any questions on this or any other topic. But thank you.
Okay, thanks, Steve. That was a great seminar. Lots of really exciting research. As a reminder, if you're not on the mailing list to receive invites to these science seminars and you'd like to be added to it, or if you or your team has work that you'd like to share with us at an upcoming science seminar, please contact us at the Office of the Chief Scientist. Our email address is aep.ocs@gov.ab.ca. And with that, I think there's been such a great presentation today. So thanks again, Steve, for sharing this all with us. Thanks to those of you who tuned in, and we'll see you all again next time.
Thanks, everyone, for listening. I appreciate it.